Good morning, everyone. Thanks first to the JGI for the opportunity to talk to you today about my work with Tiny Earth and promoting plant health. So we're interested in this for a very simple but important reason, and that's pathogens damage crops, which leads to less food that we can eat. And pathogens damage actually 16% of the global crop yield. And pathogens such as Erwinia showed here can cause bacterial wilt. And so the bacteria grows to such high density in the plant that when you cut the stem, the bacteria literally oozes out. And so this is just one example of the types of diseases that pathogens can cause to our crop production. And so this is only going to get worse in the presence of climate change. So what I'm showing here is the delicate balance between a healthy plant on the right and a sick plant on the left. And you can see that environmental conditions affect the success of the pathogen. So in the presence of climate change, we know there will be increased temperatures and increased rainfall. That's going to push the arrow towards disease. So we're looking at earlier outbreaks and more outbreaks. And so what we need is something to tip the scales back in our favor, and to prepare for the increased disease burden brought on by climate change. And so one way to do this is through the development of novel antimicrobials specifically for the use in agriculture. And today I hope to discuss how Tiny Earth is poised to address this question and how genomic sequencing is going to play a pivotal role. So the agenda today is to first talk about what is Tiny Earth and what we hope to accomplish. And second will be our specific efforts into identifying those novel antibiotics. So first, what is Tiny Earth? It started as an introductory microbiology course developed by Dr. Joe Handelsman to inspire and retain students in STEM through the thrill of independent laboratory discovery. And as the name suggests, Tiny Earth, the students are looking for microorganisms in the soil that produce antibiotics. And so the product of this discovery, a potentially new antibiotic, could actually help people and society. And so working on something greater than themselves really fuels the enthusiasm of our students. And so Tiny Earth addresses two very real problems. And the first is the antibiotic crisis. You can see that the discovery of novel classes of antibiotics came to a screeching halt in the 90s. And so this, coupled with the spread of antibiotic resistance, is leading to a crisis where our supplies of effective antibiotics are dwindling. And so we know a lot about the antibiotic crisis in humans, but what about plants? What about agriculture? So our arsenal of antibiotics to treat disease in plants is very small. There are only 3 approved antibiotics for use in disease management in plants, and 2 of them are also used in humans. So I think this is an area, especially with the increased disease burden due to climate change, is an area of research that we need to give our attention to. And the second problem that Tiny Earth addresses is the STEM deficit. So a 2012 PCAST report to President Obama showed that we are going to be 1 million STEM graduates short over the next decade to meet the job market demands. And one reason is 60% of declared STEM majors actually leave before they graduate. So Tiny Earth aims to use discovery-based course design to keep students interested in STEM and graduating with these STEM degrees. So where do students actually get these antibiotics? And of course, they go to nature's pharmacy, the soil. So the students will go out into their communities, maybe engage with their neighbors or farmers, teach them a little microbiology, get their soil sample, and bring it back to the lab. The soil is then plated for microorganisms. The microorganisms are isolated for pure culture, and then the organisms are tested for antibiotic production. And we do this through a spread patch assay. So the pathogen to be inhibited is spread on the plate, and then a soil microbe is patched on top, and then after about 24 hours of growth, we are looking for zones of inhibition, or the clearing around the soil isolate that suggests an inhibitory interaction is occurring. So you can see that the orange isolate in sort of the top right is not inhibiting the pathogen, but all of the rest have those zones of inhibition. And so we select the active isolates for chemical analysis to try and identify this, the antibiotic mediating the inhibition. So we have a broad screening panel, and they're all safe relatives. We're trying to keep this at biosafety level 1 for the students. These are safe relatives of pathogens notorious for gaining antibiotic resistance. And we include both gram-negative and gram-positive to immediately determine the breadth of antibiotic production as broad, inhibiting lots of pathogens, or narrow, inhibiting 1 or 2. And importantly, we can prioritize agriculturally relevant isolates by focusing on the inhibition of plant pathogens such as Erwinia and Pseudomonas. And we also have Saccharomyces cerevisiae as a stand-in for fungal pathogens. So, I've told you that Tiny Earth uses the rich microbial diversity of the soil coupled with hundreds of classrooms of budding scientists eager to discover the next antibiotic. But how do we turn our collection of antibiotic-producing microbes into an actual molecule? So the second half of my talk, I'll first start with some dogma in the industry around antibiotic discovery and how we think Tiny Earth's network, chemistry hub, and the collaboration with the JGI is going to overcome some of those challenges. So first, there's a little bit of dogma in the big pharmaceutical companies, and it's that the soil is tapped out. All of the most abundant antibiotics have already been discovered, and it's not really worth screening anymore. And a second problem is that the return on investment for antibiotic development is very low. So it can cost hundreds of millions of dollars to bring a safe and effective antibiotic to market, but then the product is only prescribed for like a week, so it's not bringing in the returns that something addressing a chronic illness would do. And so for these reasons, most big pharmaceutical companies have shut down their antibacterial divisions. And honestly, based on how things are, you can't really blame them. It's not a great business model. And so it's opened up space for the public sector to stand up and start making these antibiotic discoveries. For example, in the Handelsman lab, there have been multiple antimicrobial discoveries from soil microbes over the years. And these discoveries are the inspiration for the Tiny Earth course. And so we truly believe we will be successful in our natural product discovery. And the first and foremost reason is our network. So Tiny Earth is taught in 45 states and 15 US countries. And that's about 10,000 students per year taking Tiny Earth. And so the sheer scale of isolates screened and the geographic soil diversities tested is crucial for our success in antibiotic discovery. And our success is strengthened by the fact that we're interested in narrow spectrum isolates. So these molecules were discarded for decades because, again, the return on investment wasn't as big as a broad spectrum antibiotic. And so we can use our broad screening panel with multiple pathogens to find those narrow spectrum antibiotics. And so for these reasons, we think we will achieve our goal of antibiotic discovery. And so again, we're building this collection of antibiotic-producing microbes, but how do we actually get a molecule? And so this past year, Tiny Earth has established the chemistry hub to move into the next phase, which is centralized antimicrobial characterization. And so chemical analysis of our strains is one of the more expensive steps of the protocol, and it's not accessible to all of our instructors. So Tiny Earth developed the chemistry hub, so we invite instructors to send us our strains, and we'll do the chemical analysis for them. And so we have almost 500 strains in our collection right now, and it's only been operational for a semester. So that demonstrates the popularity of this service within our network. And so briefly, all that's done is a isolate is grown overnight, and the molecules are extracted in various solvents. We don't know the chemical structure of the antibiotic, so we have to try different solvents. And then the extracted molecules are tested again for inhibition of the pathogen. And in this example I'm showing, there's a huge zone of inhibition with ethyl acetate. So hopefully the molecule mediating this inhibition is found in the ethyl acetate culture extract, which is then run over a mass spectrometer to determine the chemical structures or the masses within that extract. So this is really useful for helping us, nail down the identity of this antimicrobial. But there are some downsides. One, the molecule might not be able to be extracted in the common solvents, and two, the number of molecules in the ethyl acetate extract could be up to 100 or hundreds of molecules. And so separating them out is a laborious task to find the one that's mediating the antibacterial action. But if we had an idea of the type of molecule we were looking for, it would greatly aid in its discovery. And that brings me to our collaboration with the JGI. So Tiny Earth and the JGI have teamed up to sequence some of our Tiny Earth isolates to try to identify the biosynthetic gene clusters that could be responsible for making our antimicrobials. So through the community science program, we've done a pilot screen of 29 isolates found in my home state of Wisconsin. So here we are. The upper right corner is students isolating bacteria in the fall. I wisely waited until summer. You can see Lake Mendota in the background, and I just wanted to say that this is my first field work assignment as a scientist, finding bacteria to sequence my first genome. So I'm really grateful for this opportunity to sort of leap into genomics. And here's our collection of strains to be sequenced. And at first glance, you can see that they look different. There's different colors, different extra polysaccharide production, different sizes. But in fact, the phylogeny was rather narrow. So the only gram-positive genus we found was Bacillus, and Bacillus amyliquafaciens was isolated 5 times. And 2/3 of our isolates were Pseudomonas, with 4 weird ones, Erwinia, Rhinella, a possible new Citrobacter, and Butioxella. And so different strains within the same species are indicated by the capital letters. And so I'm showing the antibiotic production profiles as a heat map, with red being strong inhibition and white being no inhibition, based on the zones of inhibition assay in the spread patch plate. And the different columns are different pathogens that are inhibited. So you can see that we have a broad spectrum of types or levels of antibiotic production. Some are very broad, inhibiting lots of pathogens, and some are narrow. And some of our broadest inhibitors are 2 of the Bacillus amyliquafaciens strains, Rhinella, and a couple Pseudomonas, Donghunensis, Vranovensis, and one of the fluorescents. And then the spectrums of inhibition vary all the way down to Bacillus simplex, which can only inhibit Bacillus subtilis. At the very top of the phylogeny. So you may notice that we had some repeats. So we isolated the same microbe a couple times. And so one of the questions we had before moving forward was, is it worth sequencing all of them? That was a question we wanted to ask because it was going to affect our Tiny Earth workflow in the future. So we wanted to include them to see how different or similar their genomes actually were. And once I answer that, I'll go into my efforts to identify novel biosynthetic gene clusters. So the first example is Pseudomonas fluorescens, and I'm showing JGI 9 and JGI 8, which have 100% 16S sequence identity. But when we sequence them, and I'm showing a pangenomic comparison, and black is simply the presence of a gene cluster and gray is the absence, that these 2 genomes are very similar, but they're not identical. There are gene clusters that are exclusive to either organism. And this actually might have been predicted by the fact that their antibiotic production profiles are different. So JGI9 can actually inhibit 2 extra pathogens, E. coli and E. aerogenes. So one would hypothesize that maybe we should look for biosynthetic gene clusters specific to those 2 organisms in the gene clusters exclusive to JGI9. Or maybe in these divergent genes that are shared by the 2 organisms, but the amino acid sequences are different. So maybe JGI8 lost the ability somehow. So this is an example of 2 strains with 100% 16S sequence identity, different antibiotic production profiles, and non-identical genomes. The second example is Pseudomonas fredericksburgensis, JGI 25 and 2. So these 2 strains actually have the same antibiotic production and 99.93% 16S sequence identity, but they have, again, exclusive gene clusters for either strain. And so the last example, I'll go back to Bacillus amyliquafaciens, which was identified 5 times. And so we had 2 white strains, and 3 JGI4 and JGI5 are white, and 3 strains that had a gorgeous orange color, JGI 6, 26, and 28. And so a little bit of history about these strains is that JGI 5 was actually identified as a white variant from a pure culture of JGI 6. So I was interested to know, is JGI 5 going to be more similar to its parent, JGI 6, that's orange, or is it going to be more similar to its white counterpart? And even just looking at the genome sizes, you may be able to predict what's going to happen. So JGI 5, the white variant, is more similar to its parent, and actually all of the orange strains look rather similar. JGI 4, the white one, is the most different. And color is not the only difference between these 5 strains. Bacillus amyloliquefaciens inhibits a lot of our bacteria in the screening profile, but only 2 inhibit Bacillus subtilis, and those are the white strains, JGI 4 and JGI 5. And so one would think that maybe I should look for the genes that are conserved in the white strains to maybe find where the gene cluster is that inhibits Bacillus subtilis. And it's this tiny little sliver right here. So I want to find those gene clusters and potentially find a biosynthetic gene cluster specific for Bacillus subtilis, or maybe simply a regulatory element that's controlling both pigmentation and or antibiotic production. And I find it actually very interesting that the loss of pigmentation is tracking with the ability to inhibit Bacillus subtilis. And I would really love to know why, and I'm not going to tell you today, unfortunately, because I'm still analyzing the differences between these genomes. Okay, so back to my original question, I would argue that yes, it is indeed worth sequencing strains with identical 16S sequence identity, especially if they have different antibiotic production profiles. So now let's get into the meat of the talk, identifying novel biosynthetic gene clusters. So the workflow I've developed is I start with the PacBio sequence, and I first want to look for whether or not the organism can inhibit another bacteria through a contact-dependent mechanism. So either with type 6 secretion systems or maybe the contact-dependent toxin-antitoxin system. And that's simply just through blasting for homologs and genes in those systems. And so if the organism contains these systems, this would be a good candidate for potentially biocontrol, adding the whole bacteria back to the plant for disease management. But they're not ideal for molecular or developing a novel molecule. So for strains that don't have these, and especially with the ability, if the chemical extract can't pull out an active molecule, both of those things together are indicative of a biocontrol candidate. So for the molecule candidates, if I'm lucky enough to have genomes of similar organisms, like I showed you before, or maybe I'll have a bunch of genomes that have identical antibiotic production profiles. So in those 2 cases, it would be really useful to use a pangenomic analysis through a program called ANVIO to narrow down the segment of the genome that I want to study. Oops. And the important part is actually identifying these biosynthetic gene clusters, the BGCs. And through that, I use a program called anti-SMASH. And it gives me a list of the biosynthetic gene clusters in an organism, and then I really want to discover novelty. We really want a new antibiotic. To do that, we compare our biosynthetic gene clusters to a curated database of known clusters through a program called BigScape, and we can get a quantitative index about how new this gene cluster is. And so I've started this process, and I'm going to give you a couple examples of biosynthetic gene clusters we found right now. So the first is a proof of principle. So we're going back to our old friend, Bacillus amyloliquefaciens. So running the genome through anti-SMASH identifies 30 biosynthetic gene clusters. And the one I'm showing here is a non-ribosomal peptide synthesis cluster, or NRPS, which is a classic way for bacteria to make an antibiotic. And so when you blast this cluster against all sequence strains, you see it's well conserved within Bacillus. And when you blast it against, for homologous genes in known clusters, 100% of the genes map to a cluster called bacilline. So I would suggest that this cluster is making an antibiotic called bacilline that has activity towards both gram-positive and gram-negative organisms, which is what I saw in the Tiny Earth screening profile. and it inhibits through protein synthesis inhibition. So it's a classic mechanism of action. So this is just to show that we can take genomes, feed them through this pipeline, and find an antibiotic-producing cluster. But what about novel BGCs? So to start, I decided to look at Butioxella, which seemed very exotic. I had never heard of it. So it was understudied, giving the likelihood of discovering something new to be increased. It is found in soil and in the ocean, and doing a literature search, I found one paper where this microorganisms made a antimicrobial, and it was a glycolipid surfactant. So it seems rich for discovery. And importantly, it inhibits both Erwinia and Pseudomonas. And so feeding the genome through anti-SMASH, I discovered 21 BGCs, and this is the only NRPS cluster in the organism. And when you blast it against sequenced genomes, you find it's well conserved in gram-negative organisms. It's found in Vibrio, it's found in Citrobacter. But when you blast it against genes in known clusters, the homology is very weak, again hinting that we found something new. So to find out what the cluster was making, I dove into the genes to see what I could find, and I found a TON-B receptor. So TON-B takes up iron siderophore complexes into the cell. And so iron is a scarce and really necessary element for bacterial life. So I would suggest that this cluster is making a novel siderophore. And for you NRPS-minded folk in the audience, there are two A domains, DHB and SIR, making a core structure of the molecule to look a little something like this. And my next example is another cluster from Butioxella that when you map it towards known clusters, maps to aryl pollenes, or APE, and these are clusters that typically make pigments. But when you blast it against sequence strains, you find that the EPE gene clusters are only half of the story. The first half of the cluster is found in a completely different cluster in a different organism. So this is the only example where these two clusters are linked, suggesting that maybe it's making a hybrid molecule, potentially with a function beyond pigmentation. So those are three examples, and I want to bring you back and remind you that we started with tiny earth antimicrobial producing isolates. With the help of the JGI, we sequenced 22 genomes and found 824 biosynthetic gene clusters. And I showed you three. So there's a lot of work to do. And so here's a classification of all of the biosynthetic gene clusters I found. And so my goal now is to start systematically working through these gene clusters and identifying which are actually making antibiotics. Okay, so if we zoom out a little further, I really think that this is an example of where we're stronger together. So Tiny Earth is providing the people and the antimicrobial isolates, or antibacterial producing isolates, and also a huge database that is continually updated with metadata of where and when these microorganisms were found. The chemistry hub is producing chemical properties and potential masses of the molecules, and the JGI is providing sequences and enzymes and the chemical space that these bacteria are capable of making. And so together we can get rid of all of the extraneous information and really nail down the goal, which is a new antimicrobial. So I hope today I've convinced you that Tiny Earth is a powerful crowdsourcing model able to fill the vacuum left by pharmaceutical companies leaving antibiotic discovery. Also, that promoting plant health with antibiotics, especially in the coming storm of climate change, is an area that we should be devoting our time and energy towards. And Tiny Earth can address this by focusing on inhibition of plant pathogens like Erwinia, Pseudomonas, and then Saccharomyces, which I didn't really talk about today, but it is in our pipeline. And finally, it's the synergy between the chemical and genomic analysis that's really going to speed up this discovery. So where do we go from here? So there's a couple points that I want to make that we're thinking about, and the first is to really focus on narrow spectrum isolates. So I think this is our greatest likelihood of discovering something new, and when these products are brought to market, it would reduce the spread of antibacterial resistance. Second, you may remember the narrow phylogeny of our pilot screen. So to increase phylogeny, we have a couple ideas. One is to include Actinomyces media. The second is to simply just increase incubation time of our soil isolates and allow for the slow growers to form. And finally, I work in the Handelsman lab, so we're interested in microbial communities. And we know that some phenotypes are dependent on interbacterial interactions. So we're actually developing a co-culture method to integrate into the Tiny Earth protocol to stimulate some of those cryptic biosynthetic pathways. And I'd like to highlight one last time all of the students and all of the instructors who are out there doing the work, finding the antimicrobial producing isolates. They truly are the heart of the organization. And if anyone is interested in bringing Tiny Earth to their institution, please come talk to me. Okay, with that, I'd like to thank the entire Tiny Earth network, my lab, the leaders of Tiny Earth, Nicole Broderick and Sam Rickers, and also Nicole Shapiro and Nigel Mounsey, who have been very supportive and helpful as I transition into studying genomics. So, thank you.
Hi, thanks for that talk. I have a question about plasmids in the genomes that you're getting back and whether you've explored whether your biosynthetic gene clusters are on plasmids or genomes, or how you're dealing with trying to figure out whether those traits are potentially transferable.
Yeah, that's a great question. We have thought about that, and one of the ways we're looking at it is whether or not the biosynthetic gene clusters are on different contigs. So we have, most of them have one contigs, but a couple have multiple. So we're thinking that maybe those could be mobile elements.
Hello. I was from Madison, Wisconsin, actually. I know Handelsman lab and doing, it's already invented this called metagenome approach. The major argument is the culture approach. You only can explore maybe 1% or less than 0.1% of the soil microbes. So I know I don't know if you thinking about that, how we can, advance the metagenome technology, even make the antimicrobial discovery more efficient than, repeat the big pharma already explored. Big pharma was doing the scale just unbelievable, and yeah, that's my comment.
Mm-hmm. I think that's a great comment, and you're absolutely right that we are limited by what we can culture in this setup, but including metagenomics actually in the classroom might be a really interesting way to teach students about that exact problem. So I'll take it to Joe.
So over here,
one more over there.
This is less of a scientific question and more related to the ethos and model of Tiny Earth. So let's say you find something that is really interesting, really powerful. How do you go to market and related to IP? I mean, maybe this was isolated in a small school somewhere else outside of the U.S. What happens? Also related, like most likely right now you have to adopt the Nagoya protocol with things coming from elsewhere, right?
Right. So yeah, we have a lot of material transfer agreements that people have signed, and I think it would be great if anything came from this, that the people who actually isolated it would get a kickback. I think that would be incredible, because we aren't a company, we are just scientists. I don't think I would see any money from this if anything came of it. So yeah, I don't really know, but I hope that it would be, spread around.